Good morning. It's July 29th, and this is your daily brief in science. Here's everything you need to know. Researchers in Philadelphia have made a significant advancement in the fight against Alzheimer's disease with a new blood test that greatly improves both the speed and accuracy of diagnoses. A recent study conducted in Sweden, involving over 1,200 patients, revealed that this blood test achieved an impressive accuracy rate of 91%. This performance far exceeds the 61% accuracy of primary care doctors and 73% accuracy of specialists in diagnosing the disease. If this blood test sees widespread adoption, it could drastically reduce the wait time for patients to qualify for Alzheimer's treatment, shortening it from nearly 6 years to a range of 6 to 13 months. The test incorporates biochemical markers, particularly p-tau217, which correlates with amyloid plaque buildup in the brain, making it a strong indicator of Alzheimer's pathology. Experts, including Professor Jianfeng Feng from Warwick University, have referred to this blood test as a potential breakthrough that could be integrated into routine screening practices. Despite the promising results, the broader implementation of these blood tests will necessitate further research, the establishment of guidelines, and education for healthcare professionals. Currently, over 6 million Americans are living with Alzheimer's, amplifying the need for early and accurate diagnosis. Traditionally, diagnosing Alzheimer's has relied on invasive techniques such as brain scans or spinal taps, which are not always readily accessible. Another similar test for p-tau217 has demonstrated even higher accuracy rates, achieving up to 96% accuracy in identifying elevated beta amyloid levels. This advancement is part of ongoing research efforts aimed at enhancing diagnostic tools for neurodegenerative diseases, which involve complex mechanisms like neuroinflammation and oxidative stress, complicating the diagnostic process. Paradromics is making strides in the field of brain-computer interfaces, or BCIs, by developing technology that translates brain signals into speech. Founded in 2015, the company has successfully raised $87 million in venture capital and $18 million in public funding. Their device has received 2 FDA breakthrough device designations, marking significant progress in its development. Additionally, Paradromics has been accepted into the FDA's Total Product Lifecycle Advisory Program, which will assist them as they prepare for human clinical trials slated to begin next year. Recent feasibility tests involving 10 subacute stroke patients have shown promising results, with attention classification accuracy ranging from 70% to 100% during online assessments. The BCY system operates reactively, measuring brain responses to external stimuli, which is particularly beneficial for enhancing sensory rehabilitation in stroke survivors. Current sensory training methods primarily employ bottom-up approaches, utilizing various tactile stimuli and electrical stimulation to aid recovery. Non-invasive brain stimulation, or NIBS, has emerged as a potential adjunct treatment for stroke rehabilitation, particularly in improving motor function in the upper limbs. Given that stroke is a leading cause of disability worldwide, affecting 50% to 85% of survivors with sensory deficits, effective rehabilitation interventions are critically needed. A review of NIBS highlights the necessity for more randomized clinical trials to clarify the effects of contralesional stimulation and identify suitable biomarkers for patient response. Further research is required to optimize NIBS protocols based on individual patient characteristics, such as the integrity of corticospinal tracts. Clinical assessments conducted on study participants established baseline functionality, including evaluations of motor and sensory scores. Interestingly, a gender-based analysis revealed that men showed a 9.16% greater improvement in results compared to women, indicating a need for further investigation into gender-specific performance differences. A new hardware device named CRAM has been introduced by researchers at the University of Minnesota, promising to revolutionize energy consumption in artificial intelligence applications. This innovative chip has shown the potential to reduce energy usage by at least 1,000 times, with estimates suggesting savings could reach up to 2,500 times compared to traditional computing methods. CRAM leverages spintronic devices known as magnetic tunnel junctions, or MTJs, to execute computations directly within memory. This approach effectively removes the need for energy-intensive data transfers, addressing a significant bottleneck present in the conventional von Neumann architecture. As a result, CRAM is positioned to enhance energy efficiency in AI technologies, a critical development given the rising energy demands of these systems. The International Energy Agency projects that global energy consumption for AI will exceed 1,000 terawatt-hours by 2026, more than doubling from 460 terawatt-hours in 2022. This alarming forecast underscores the urgent need for advancements like CRAM to mitigate energy consumption in AI applications. Jianping Wang, the senior author of the study, highlighted the importance of interdisciplinary collaboration in bringing this technology to fruition, with contributions from researchers at both the University of Minnesota and Arizona University, supported by various U.S. government agencies. The foundational work for CRAM has been in development for over 2 decades, with initial concepts dating back 20 years. The research findings were published in the peer-reviewed journal NPJ Unconventional Computing, and the team holds multiple patents related to the technology. Moving forward, the researchers aim to partner with semiconductor industry leaders in Minnesota to demonstrate and produce hardware that will further enhance AI capabilities, addressing challenges in scalability, manufacturing, and integration with existing technologies. Drought is emerging as a serious challenge to global food security, exacerbated by climate change's impact on agricultural practices. Understanding how plants respond at the molecular level is crucial for developing effective mitigation strategies. Recent studies highlight the role of DNA demethylases in regulating gene expression, stress responses, and overall plant development, which is vital for crop improvement. Epigenetic modifications offer a rapid and reversible mechanism for plants to adapt to stress, allowing them to respond without permanent changes to their DNA sequence. The concept of epigenetic memory is particularly important in drought-adapted plants, where DNA methylation patterns established during initial drought stress can persist, influencing future responses. MicroRNAs, or miRNAs, also play a significant role in regulating gene expression after transcription, interacting with epigenetic pathways to facilitate chromatin remodeling. Advanced technologies, including bisulfite sequencing and CRISPR-Cas9, are set to enhance our understanding of plant responses to water deficits, paving the way for the development of drought-tolerant crops. A recent systematic review identified 401 studies focused on epigenetic mechanisms in plants, with Arabidopsis thaliana and Oryza sativa being the most researched species. However, there is a notable gap in research on non-economic and tropical plant species, indicating a need for broader investigation. China is leading in published studies on this topic, supported by robust government investment in scientific research. Over the past decade, there has been a marked increase in epigenetic studies, with around 90% of research conducted recently. Most studies have concentrated on plant responses to abiotic factors such as temperature, water availability, and salinity, while less attention has been given to light. Additionally, protein structure analysis has revealed conserved domains, and gene duplication analysis has underscored recent whole genome duplication events that contribute to gene diversification. Methylome DNA sequencing remains the primary technique for detecting epigenetic interactions in current literature. As humanity sets its sights on deep space exploration, genome editing is emerging as a potential solution to help astronauts adapt to the harsh conditions they may face. Experts are investigating how gene modifications could safeguard astronauts from the significant health risks associated with space radiation, which includes an elevated risk of cancer. Research is currently underway to explore the possibility of transferring genes from tardigrades, organisms known for their extreme resilience, into humans. This could significantly enhance survival rates in the unforgiving environment of space. Additionally, potential applications of gene editing may involve inserting radiation-neutralizing genes from plants and bacteria into human DNA, as well as engineering crops to endure radiation exposure. However, the rapid advancement of gene editing technology is outpacing the ethical discussions surrounding its use, leading to an uncertain future regarding human adaptation in space. Concerns regarding the permanent alteration of the human species are fueling opposition to genome editing technologies. The global controversy surrounding the 2018 creation of gene-edited babies by Chinese scientist He Jiankui brought these ethical dilemmas to the forefront. Jiankui faced legal repercussions for his premature attempts at gene editing, which ignited significant ethical debates within the scientific community. A recent discussion featuring Matt Honan and Antonio Regalado with He Jiankui delved into the implications of this controversial technology for the future. Ultimately, the role of genetic enhancement in future space missions will hinge on societal acceptance and the establishment of regulatory frameworks governing their use. As humanity confronts the challenges of space travel and colonization, including extreme radiation, temperatures, and the effects of weightlessness, the regulatory approaches to genome editing will vary across countries, with Germany and Canada taking particularly cautious stances. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.